So on to our last trip through the digestive system with digestion and absorption. By now, you're probably familiar that digestion includes both mechanical digestion, so breaking down food doesn't change the composition, but it does make these smaller particles. The small particles are going to contain nutrients, fats, carbohydrates, and proteins, and these are going to be broken down by chemical digestion, and then they will be absorbed across the cells of the small intestine. So what is absorbed? We are going to absorb carbohydrates, proteins, and triglycerides. Starch will get broken down into glucose. Lactose gets broken down. So lactose, sucrose, and maltose are disaccharides, and they will be broken down by enzymes, lactase, sucrase, maltase, things that end in ase, into their monosaccharides. And then we'll talk about, once we get to metabolism, we'll talk about how glucose and galactose are transported, and then how fructose crosses through facilitated diffusion. So these are going to be slightly different. Okay. Proteins, we'll talk about their breakdown into amino acids, di and tripeptides. There are different approaches when we are breaking large substrates apart. And so we tend to have an enzyme that breaks apart large chunks into small chunks. Okay. So we'll have an enzyme that breaks proteins into polypeptide segments. Same goes with starch. And then we'll have another enzyme that breaks things off one at a time. So for carbohydrates, we have the enzymes that break disaccharides into monosaccharides. When we get to proteins, we'll also have one that just takes off one amino acid from at a time from the C terminal end, which you don't need to know, but it takes just one at a time. So it's usually a multi-pronged approach to breaking these nutrients apart. Triglycerides will get broken down into fatty acids, and monoglycerides, it's hard sometimes to take off that last fatty acid from glycerol. So that's a monoglyceride. We're going to start at the top and go towards the bottom. Same old routine. Okay, so we'll start at the mouth. The mouth is where carbohydrate digestion begins. Someone else. So carbohydrate digestion begins in the mouth. It will get cut off at the stomach, and then it will resume in the small intestine. So one of the things that you need to know is where digestion of carbohydrates occurs. Where does digestion of protein occur? Where does digestion of lipids occur? So where does it occur? And oops, there we go. Okay, so this slideshow will look slightly different from those of you who have downloaded it. I was going through it last night and was just unhappy with the slides for the rest of this and metabolism as well, which is why I only uploaded the first 23, which I don't even think we're gonna get that far. But so I will reload what we're going through today. I'll put a new file in for reference. It's not terribly different, but I don't know. I thought that we needed more summary tables, more diagrams. So sorry for the last minute change, but I don't know. If you know me, things just sound like, I don't like that, I want to change it. Okay, so I have these new summary diagrams in here. This one's at the end of carbohydrate digestion before we start to protein, and then we have one for fatty acids and nucleic acids. But I do really like the material, how it is. These come from your book, so they're also in your book, but I like how it's organized into one place. And it actually, so it really includes everything that maybe not everything, the 80-20 rule, most of what you need to know for the exam. So for carbohydrate digestion, you need to know where it occurs. So it's going to start in the mouth. Then we'll go through each of these steps in more detail. I just wanted to show you that we do have these little summary tables now that are a good point of reference. Okay, so it starts in the mouth. It will not occur in the stomach, okay, because the stomach acid breaks apart that enzyme. Amylase is the enzyme. So we start with salivary amylase, and it's going to get destroyed by the stomach. It does not, you know how, so enzymes have that optimal pH that they work in, and a pH of 2 does not work for amylase. You need a more neutral-ish pH. So begins with salivary amylase. To break down those carbohydrates, it stops in the stomach because salivary amylase gets denatured and cannot continue. Then it'll pick back up in the small intestine after we get that pH back towards neutral with pancreatic amylase. And then the final step is going to be with these brush border enzymes. Brush border enzymes are literally secured to the lining, the apical membrane of these simple columnar epithelial cells. So brush border enzymes, they're going to stay attached, and then they will do that final step of breaking down disaccharides to monosaccharides. Okay. So now we're going to go back to all of these little processes in details. All right. Okay, so carbohydrates, that digestion starts in the mouth with salivary amylase. Salivary amylase will break down larger carbohydrates. So it starts that carbohydrate digestion by taking starch, which is chains in their branch change of thousands upon thousands of glucose molecules. And then the salivary amylase is going to make it into oligosaccharides. So disaccharides are 2, and then up to a chain of 8 glucose molecules. So these are much smaller chunks that then when they get through to pancreatic amylase, that the pancreatic amylase can work on. And then the brush border ones will work on the disaccharides once they get to that level. As I mentioned, salivary amylase is inactivated by stomach acid and digested by stomach proteases. So it's inactive and then it's eaten up. So it's only going to work in the mouth and then as it's moving down the esophagus. So it gets things started, but then it'll pick up later. The body only digests 2 types of polysaccharides, so starch and glycogen. These are just chains of glucose molecules. One is produced by animals, that is glycogen. So if you're eating a burger, you'll ingest glycogen from that meat. And then starch, so potatoes, corn, are going to have a lot of starch molecules. We cannot break down another polysaccharide called cellulose. We don't have the proper stomach structure or the proper enzymes. So cellulose takes a long time to digest. We would spend all day eating, grazing, but it does become fiber, which increases the bulk of the stool and helps move things through the GI tract. A low fiber diet increases the risk of colon cancer because the waste starts to sit in the colon for too long. So you want to have enough fiber in your diet to move the feces along. Okay, so we can digest starch, which is made by plants, and glycogen made by animals. We cannot digest cellulose. Now we're going to make our way to the stomach, and we're going to stop carbohydrate digestion because amylase gets inactivated and digested. But we're going to pick up protein digestion. So protein digestion starts at the stomach. We have, does anyone remember what type of cells release pepsinogen, sorry. We have 3 cell populations in those gastric glands. Actually, we've got 4 cell populations. So we have enteroendocrine, chief cells, parietal cells, and mucous neck cells. Does anyone remember which one produces pepsinogen? So it's
Is it chief cells?
It is, yay, it is. Okay, so chief cells produce pepsinogen, and then which cells secrete hydrochloric acid?
Parietal.
The parietal. So we have those gastric, oh, you're in the chat, sorry. Okay, I didn't have that one up. I'm gonna put that one up now because I might not see more questions. Okay. So we have those chief cells, they secrete pepsinogen, they pass by the hydrochloric acid being produced by the parietal cells, and that converts pepsinogen into pepsin. Pepsin preferentially breaks the bonds between tyrosine and phenylalanine amino acids. This will then, so by picking those 2, it means it's going to break polypeptide chains into smaller polypeptides, okay? But some are going to be longer, some are going to be shorter, but it does break that particular linkage. By breaking that particular linkage, the polypeptides get smaller, and then pepsin, again, we have that optimal pH for each enzyme, and pepsin prefers a pH of about 2. So it actually gets denatured when it enters the duodenum. So pepsin is only going to be active in the stomach, but we've broken amino acids into much smaller polypeptide fragments. And then we are going to, the bulk of digestion and absorption occurs in the small intestine. And the small intestine is actually the major organ of the digestive system. We tend to think of the stomach because it rumbles and I don't know, gives us heartburn. Like we just pay more attention to it, but it's really the small intestine that is the workhorse of the digestive system. Carbohydrate digestion is going to pick back up in the small intestine. That we now have pancreatic amylase, and that is going to continue to work on starch and on polysaccharides to break those down. And then we have those brush border enzymes. There are 2 categories. Dextrinase and glucoamylase break down oligosaccharides. So anytime you see the word amylase, so whether it is salivary amylase, pancreatic amylase, glucoamylase, amylase breaks down oligosaccharides. The previous word just tells you where you're going to find it. If you're going to find it in saliva, if it's going to be made by the pancreas. And then this one, not super helpful, but it is the last location, and that is the brush border. Then these 3, I think the enzymes pretty much make sense. Lactase breaks down lactose, sucrase breaks down sucrose, maltase breaks down maltose. So These are in, we've got glucose and galactose, glucose and fructose, glucose plus glucose, or the monosaccharides. It's kind of from last semester. Individuals that are lactose intolerant actually don't have the enzyme lactase. So absorption, only monosaccharides can be absorbed by the epithelial lining of the small intestine. So if an individual is missing lactase, then the lactose can't be broken down. And so the lactose, it's going to also hold on to water, but it's going to cause bloating and diarrhea because it can't get broken down and reabsorbed and absorbed. So we have dextranase and glucoamylase breaking down oligosaccharides into disaccharides. Okay, so we have polysaccharides, oligosaccharides. So these are 2 to 8, really 3 to 8. Otherwise, it wouldn't be needing to be broken down. So they end up as disaccharides, and then the disaccharides are going to get broken down by the brush border enzymes, which are located right next to either a channel that's going to allow for co-transport or a channel that's going to allow for facilitated diffusion. Fructose is the least common monosaccharide in our normal diet. So an industrial westernized diet has kind of messed things up, but if we were to go back to hunter and gatherer, we really wouldn't have a tremendous amount of fructose in our diet. So fructose travels via a different mechanism. So fructose is going to be absorbed into the intestinal cell by facilitated diffusion. It's going to travel down its concentration gradient. That's because we don't have very much fructose, and so it just moves right along and then it'll get transported out. So fructose, facilitated diffusion. Glucose and galactose are very similar biochemically. All 3 of these are C6H12O6. So they have the same number of carbons, hydrogens, and oxygens. It's just how they're arranged in a ring structure. Glucose and galactose are absorbed or transported into the intestinal cell by sodium co-transport. So this is secondary active transport using sodium ions as that energy source, that sodium ion concentration gradient as that energy source. Okay, so fructose, facilitated diffusion, that's a passive process. Glucose and galactose use secondary active transport to get into the epithelial cell. This goes over how pancreatic amylase breaks down starch and glycogen into oligosaccharides up to about 8 carbons long, and then disaccharides. Then we have 2 categories of brush border enzymes. We have glucoamylase, which is going to break down oligosaccharides, and then we have lactase, sucrase, and maltase that are going to break disaccharides into monosaccharides. Then if it is fructose, it'll be transported by facilitated diffusion. If it is glucose or galactose, it will be transported using secondary active transport, co-transporting as a symporter. As the sodium comes in, the glucose or the galactose will also come in. And then we haven't gotten to this point yet, but monosaccharides exit the basolateral membrane by facilitated diffusion and enter the capillaries via capillary clefts. Okay. This is, like I said, this is our new summary table. Carbohydrates start in the mouth with salivary amylase. That gets digested and inactivated in the stomach. We pick up with pancreatic amylase, and it is going to break starch and glycogen into oligosaccharides and sometimes disaccharides. And then we pick up with the brush border enzymes that convert disaccharides into monosaccharides. And then these monosaccharides are transported either via facilitated diffusion for fructose or secondary active transport for glucose and galactose. And then these will be transported across that basolateral membrane. We spend more time on breaking them down and getting them into the cell, and then we don't worry as much about them getting across the basolateral membrane, except for when we get to lipids. That's going to be a whole new ball game. Okay, so for proteins, the following slides are going to discuss this in detail. Protein digestion is going to start at the stomach with pepsinogen that gets released and then activated to become pepsin, and it will break at those specific locations into polypeptides. Pepsin gets inactivated at the stomach, and pancreatic enzymes trypsin, chymotrypsin, and carboxypeptidase will pick it up, and then as well as and these have different mechanisms that we'll talk about. And then the brush border enzymes. So again, we have 3 locations. So for the stomach, we had pepsin, and it was going to break polypeptides into smaller polypeptides. Then it gets inactivated at the level of the pancreas because it doesn't like a pH that's closer to neutral. So then we need pancreatic enzymes. So for pancreatic enzymes, we have 2 categories. We have trypsin and chymotrypsin, which are going to break the polypeptides into smaller polypeptides like dipeptides and tripeptides. Then we have carboxypeptidase, which will peel off 1 amino acid at a time. So it just takes 1 off at a time. Then the brush border enzymes are going to act. We have dipeptidase, which is going to act on small peptides, and then aminopeptidase, which is one amino acid at a time. So you're probably wondering, like, how many of these are you going to need to know? And let me go through this and I'll go back up. Okay, so absorption, dipeptides, tripeptides, and amino acids. So you don't have to get all the way down to amino acids on this one. Will also be transported by secondary active transport with sodium. Dipeptides and tripeptides will be broken down inside the epithelial cells before getting transported across that basolateral membrane. So pancreatic proteases break down proteins and protein fragments into smaller peptides, and some of those will be individual amino acids because we have carboxypeptidase. Then we get to the brush border enzymes, which will further break down polypeptides into tripeptides, dipeptides, and amino acids. If they do make it across, then they're co-transported through secondary active transport with sodium. If they are transported as a di or tripeptide, those will get broken down inside the cell and then transported across that basolateral membrane using facilitated diffusion again. And before we get to lipids, because lipids are different, kind of going back to what do you need to know, right? Okay, so you do need to know the site of action. Does it occur in the mouth? Does it not occur in the mouth? Does it occur in the stomach? Does it not occur in the stomach? Small intestine, yes, no. Small intestine is pretty much usually yes for just about everything, but they need to know if something is digested or not in each location. Then how many names do you need to remember, right? So salivary amylase, you do need to know that one specifically is going to be breaking down carbohydrates in the mouth. Same as pancreatic amylase. For the brush border enzymes, you don't need to know dextranase and glucoamylase per se, but you do need to know that there are a group of enzymes that break down oligosaccharides versus I would expect you to know these three because they match their substrates so well that these three break down disaccharides into their monosaccharides, and then how these are transported. Fructose is facilitated diffusion. Glucose and galactose are through secondary active transport. Moving on to protein, it starts in the stomach. It's going to continue with the small intestine, and then it's going to continue with the brush border section of the small intestine. However, it's not one enzyme carries through all of those. You should know that pepsin, because we've gone over it so many times, is being one of, the enzymes produced by the stomach. Well, and it's pepsinogen activated by hydrochloric acid. So that one is fair game. And then the pancreatic enzymes you should know these collectively. If you saw trypsin, chymotrypsin, carboxypeptidase, you should know that these are pancreatic protein digestion enzymes. Do you need to know if it's like carboxypeptidase, one amino acid at a time? No, but you do need to know that these three are pancreatic enzymes that break down proteins. And the same goes for the brush border enzymes, that you need to know that these as a group. So if you saw aminopeptidase, carboxypeptidase, and dipeptidase, that these belong to the brush border enzymes and are going to break down polypeptides or amino acids. Okay, so again, you don't need to know what the function is for each one of these, but collectively, you should know that these belong to brush border, that these, the trypsin, chymotrypsin, carboxypeptidase, belong to the pancreas. And then there will be several questions involving pepsin, pepsinogen, activation to pepsin. This one gets more time and attention. So in a way, carbohydrates and protein digestion, absorption, and then transport across the basolateral membrane, they have a lot in common. There's more similarities than differences. That is not true when we get to lipids. So lipids are their own animals. They are hydrophobic, and they tend to like to glob together because they're hydrophobic. So they kind of come with their own set of problems, challenges for digestion. So the first thing that needs to be done for lipids, and this is going to occur in the small intestine. A lot of it will occur in the duodenum, because if we don't emulsify them, we're not getting any further. So for lipids, it's going to require both pancreatic lipase as well as bile salts, which are produced by the liver and stored by the gallbladder. And the two, we're also going to need lecithin that's secreted in the bile. Okay, so bile is made by the liver, stored in the gallbladder, secreted through that common bile duct. And the two components that really play a role in lipid digestion are bile salts and then later lecithin. Okay, so the first thing we need to do is we need to take large globs of fat or lipid and break them into smaller pieces so that the lipase has more surface area to act on. So bile salts are polar. And so bile salts will start to pull the droplets apart to become smaller droplets. That increase in surface area allows for digestion by pancreatic lipase. So pancreatic lipase, so ACE is an enzyme, cleaves triglyceride into fatty acids or monoglycerides. So if you remember, glycerol is a 3-carbon fragment, and it's got hydrogens off of here. And then we have 3 fatty acids. So pancreatic lipase can cleave off these first 2 fatty acids. So we end up, but this third one stays attached. So we have a monoglyceride. So we end up with monoglyceride plus 2 free fatty acids. Okay. Monoglycerides. So the third step is something called micelle formation. And people are actually more familiar with this because it's micellar water, like to clean your face. And so people have at least started to hear about micelles. So when those free fatty acids are broken off, or once we get down to a monoglyceride, then bile salts are going to glob onto them, like to keep them separate. They're like, oh my gosh, we've worked so hard to like break big droplets into small droplets that we're not going to let you go backwards and coalesce again. So bile salts and lecithin quickly attract to those hydrophobic sections, and they've got hydrophilic sections too because they're polar, to form micelles. So micelles are emulsion droplets. So they're very similar to the step 1 droplets, except that they are 500 times smaller. And because these micelles are so small, they can diffuse between the microvilli to come into contact with that apical membrane. Okay, so we started off with this big glob of fat. Then we emulsified it into smaller pieces using bile salts. So these bile salts, they're hydrophobic on one end, hydrophilic on the other. So they associate with the hydrophobic end, but then they keep the other droplets away because this side is hydrophilic. Okay, so then pancreatic lipase comes in and it does its work, and we get monoglycerides and fatty acids. The bile salts and lecithin surround these quickly because they don't want them to go backwards. And now this is so small. Okay, so I'm going to draw it really tiny that it goes down to the cells, the simple columnar cells. Well, when these get next to the membrane, fatty acids are what? Are they hydrophobic or hydrophilic? Are they water soluble or lipid soluble?
Hydrophobic?
Yes, so they're hydrophobic, which makes them lipid soluble. So when they get really close to this membrane, they're just going to diffuse across it. No transporters involved. So the bile salts and the lecithin keep it essentially safe and separate. But then as soon as it comes into contact with this membrane, those bile salts and lecithin let go, and the fatty acids just diffuse across. Other cells have more complicated mechanisms, but for the small intestine, because we have they're exposed to a lot of fatty acids, they're going to, once they hit that membrane, diffuse across. So now we end up with a lot of what are called free fatty acids inside of this simple columnar cell. Okay, so on to step 4, fatty acids leave the micelles. It's literally like they hit the membrane and the bile salts pop off and they just diffuse through. So they move. So this is different. Again, everything about fats is going to be different than amino acids and carbohydrates, because fatty acids, instead of needing secondary active transport, they're going to move by simple diffusion across that apical membrane. So now we're inside the cell and we have a lot of fatty acids. Okay, so that's not going to be ideal to just like, it's kind of like, what do you do with them? So the cell is going to create chylomicrons. So the inside, the epithelial cell, we're going to take the free fatty acids and the monoglycerides and make triglycerides again. So we reform the fat into triglyceride inside the cell. Then those triglycerides are coated with lecithin and cholesterol. So we've got triglycerides on the inside, cholesterol and lecithin on the outside, and this is now called a chylomicron. And it's not just one, it's going to be many triglycerides made into these chylomicrons. Chylomicrons are big-ish. Okay, so we now have something that is big-ish trapped inside of our simple columnar epithelium. There's no way to get this through a transporter like protein, so they have to be extruded by exocytosis. Coming through the apical membrane are free fatty acids, and then these are going to be recombined into triglyceride inside the epithelial cell, coated with lecithin and bile salts, lecithin and cholesterol, sorry about that. And then these are going to be exocytosed because they're pretty large. They are going to then, now this is again something different, they enter the lacteal system. These are not going to be directly, they're not going to go in those little clefts in the capillaries like amino acids and monosaccharides, and where that absorb through into the capillary system. These things are large, and so they are going to be absorbed into lacteals, which are part of the lymph system. Eventually, the lacteals dump into venous circulation. Once they go from the venous circulation, eventually they'll make it into the systemic circulation, but they are going to be cleaved in the blood by lipoprotein lipases, which allow a small amount of free fatty acids to be kind of hanging out in the blood. And we're not going to get to binding proteins. It's a whole other category of stuff. And then in the liver, they can also be combined with proteins to make HDLs, LDLs, VLDLs, lipoproteins. Okay. You need to know the general scheme of of fat digestion. So the first step occurs in the duodenum, in that small intestine. And the first step is emulsification. So emulsification is going to require bile salts. Bile salts are polar. They're going to take this large fat globule and break it into what are called lipid droplets, smaller pieces of fat. The hydrophobic side is here, and then we've got the hydrophilic. So the bile salts keep the fat droplets from coalescing and forming a big droplet again. So once we emulsify with bile salts and create smaller droplets, then we can have pancreatic lipase is going to break the droplets into fatty acids and monoglyceride. Once we have fatty acids and monoglyceride, the next step is to form micelles. And this is going to happen when bile salts plus lecithin surround the fatty acid or the monoglyceride. Okay. This micelle is very small, 500 times smaller than the step above it. Okay, so tiny, tiny. And now we have this micelle can wiggle in between the microvilli. And when it hits this apical membrane, it'll use simple diffusion. Okay, so simple diffusion through that apical membrane. Now we get unique to, well, that's unique anyway, but we continue to get along that unique pathway. That inside the epithelial cell, we're going to reform triglycerides. So now we're building them back into what they started off with and repackage them into chylomicrons. So we're going to form chylomicrons. These are big, okay, too big for transport. So they have to be exocytosed. And they exocytose and enter the lacteal, the lymphatic system. So you need to know the steps, what order they occur, and some details-ish about what happens at each one. Okay. Then know their order, be able to also match with what's happening at each step. So we're back to these summary slides. So fat digestion, very, very unique. I didn't even talk about lingual lipase or gastric lipase because it says minor, minor. Yep, minor, minor. The major thing about fat is that it is going to take place in the small intestine. It will need emulsification, forming micelles, forming chylomicrons, exocytosing those, and then those leave via the lacteals, via the lymphatic system. So we've got bile, we've got lecithin, and we've got pancreatic lipase. Okay, for nucleic acids, that's where we're moving on to next. In the pancreas, there is ribonuclease and deoxyribonuclease. These break RNA and DNA down. Then we have brush border enzymes, which will further work on those. For nucleic acids, their breakdown is going to occur in the small intestine. So remember, if we're eating meat or plant, something else that has DNA or RNA, we are ingesting that as well. It will go into the stomach, where you'll get some breakdown in the stomach, but the main breakdown occurs at the small intestine, where we have nucleases, so pancreatic nucleases, so produced by the pancreas, but it acts in the small intestine to break DNA and RNA into nucleotides. Okay, so pancreatic nucleases break them down into nucleotides. So A's, C's, G's, and T's, remember that? Then we have enzymes, nucleosidases, and phosphatases on the brush border cells, which break nucleotides into their base and pentose sugar, and then the phosphate backbone. Okay, so it breaks it into the smaller components. The pancreatic enzymes, nucleases, break RNA and DNA into their nucleotides. The brush border enzymes break nucleotides into their components, the nitrogenous base, the pentose sugar, and the phosphate that came from that phosphate backbone. Unsurprisingly, they are absorbed into the simple columnar cell via active transport. Okay, so that's active transport is a common theme for everything but fructose and fats. Absorption of other nutrients, we go over this very briefly. Fat-soluble vitamins are absorbed along with fat micelles, which is why sometimes they'll say like take with food to avoid stomach upset, but sometimes it's also take with food because you would need the presence of fat in order to also absorb the vitamins. So if you took a capsule, let's say of vitamin E, and you just took it and you're fasted and not, so not consuming food, you're not going to absorb it because it needs to be taken with something fatty in order to be absorbed into that fat micelle and absorbed. Water soluble or absorbed by diffusion and passive and active transport. These are the B and C's. Vitamin B12 is large and charged, so it needs to be complexed with intrinsic factor from the stomach. The only thing you need to know about intrinsic factor is it's produced by the stomach and it aids in vitamin B12 absorption. That's as far as we go with that one. Electrolytes, these are going to be absorbed along the length of the small intestine. We have sodium cotransporters, potassium, sodium pumps, chloride exchangers, exchanges bicarbonate, iron and calcium. I'm not going to have a question on electrolyte absorption. I mean, other than maybe that it would occur in the small intestine. But we're certainly not going to go into the cotransport, the exchanger mechanisms. Interestingly, women have 4 times the number of active transporters than men do for iron. So just thought I'd throw that in there, even though we struggle more with anemia. And we're not going to talk about the calcium and vitamin D interaction. Okay, I feel like let's focus on carbohydrates and proteins and fats and nucleic acids, and we'll just leave the rest. Water follows sodium. 9 to 10 liters of like chyme goes through your small intestine, and usually 9 liters a day of water, and that's going to be reabsorbed. Alrighty. Okay, so that ends it for digestion.